Hello student, welcome to EPG Pathshala. I am Indira Ghosh from JONU and today we are going to talk on the module which is actually Introduction to Free Energy and Entropy of Biomolecules under the paper Computational Biology. Now, what are the learning objectives? Till now you have learned a lot about the force field, energy function, potential energy, etc., etc., solute, solvent energy, everything. Here I am going to tell you about what is free energy, because it is important to know what is free energy and how to estimate it and what are the challenges to estimate it. I also will talk about present status of calculation or estimation of enthalpic energy and a little bit about the entropic energy which are used in biological examples. Well, we should start with the concept of free energy. What is free energy? Basically, free energy is a thermodynamic quantity, it's a state variable, which represents the likelihood of a certain system conformation. That is, something like free energy F, if we call, of a system which is a canonical ensemble NVT, means number, volume and temperature, then F should be given by 1 by beta. Beta represents the Boltzmann constant and the temperature. Ln Q. Q represents the partition function of the system. When a system is defined in Cartesian coordinate, then Q actually is presented by the coordinate and momentum of the system. So, very simply, the free energy is something like a state function. But most important is that one can actually measure the free energy. So, it takes the most important space in our whole type of calculation when we do simulation, etc., molecular modeling, many conformation, ensembles, then free energy plays the dominant role in it. We are most interested in biological phenomena, the binding free energy. And what is the binding free energy? Binding free energy is something which measures the efficiency of binding between one molecule, another ligand, or two ligand binding the same macromolecule, etc. How we go about it? Now, HPR is the Hamiltonian, which we'll know, and this is actually is the sum of the potential energy and the kinetic energy over the whole space. That means how many are available. And free energy is simply written mostly by U, which is enthalpy or energy of the system, Ts, that is just the temperature multiplied by entropy. Actually, the binding free energy, its definition is that if B bind to A, then that excess energy or the energy which you release by binding into between, it may be positive, it may be negative, should be calculated in this form, FB minus FA, and the delta U represents the FB, that is how much is the energy loss or energy gain due to the binding, and how much is entropy gain or entropy loss, that is delta S. And total value should be represented by beta, which is constant with respect to time, the temperature. However, it is the ratio between available system in Q, that is QB is the ensemble or the partition function of all the ensemble in B, state B, and QA is state A. So this is the binding free energy expression. So basic component of free energy is first a suitable molecular model, and or rather I would say energy function or Hamiltonian. Then proper sampling protocol so that it can generate all possible conformers at certain equilibrium condition or room temperature, etc. Also, we require to have an estimator of free energy differences. So, which should estimate properly the free energy differences. Now we just discuss a little about molecular model. I had discussed a lot's detail about. It is nothing but an Hamiltonian which will calculate the energy and forces of a system. But most importantly, the choice of Hamiltonian should not compromise with accuracy and efficiency. However, this becomes common way of compromising. The main reason is these biological systems have a huge number of degrees of freedom. So there is a huge space available for having many conformers. However, the functional form tries to keep it within certain limit which is equilibrium, not to break the structures, etc. in terms of a layman's concept. However, basically, they are developed to keep the system in its equilibrium. So, it is not so easy to go to the spots which are least frequently are traveled. And that becomes the challenging problem while sampling is done. Now, if we look at molecular modeling already explained and the degree of freedom of a system, we actually can define the model into one is solute description, which can be done in quantum and classical. Normally in macromolecules, we use classical model. Solvent model also can be both, but we use normally classical model. But solvent model can be explicit solvent or implicit solvent. Hence, we use generally both depending on how good explanations we would like to see. Now, it is actually ultimately our model should have a solute description and solvent description. However, this will be intermingled. So, generally we like to use this modeling. What is the Hamiltonian? Classical Hamiltonian explains everything in here as it can be explained by two terms, and kinetic energy is very easy to measure because we'll have a velocity of the system. However, the potential energy is somehow defined as if actual physicochemical parameter, means internal parameter like bond length, bond angle, etc., they are parameters, plus the non-bonded like electrostatic, van der Waals, etc. Plus it can be another additional terms which we would like to add it. Hamiltonian are built like that. How it is the force field. Basically, the force field are used in physics, this word, and it is a negative gradient of a scalar potential. And chemists say it is a potential function. So the functional forms have been described much earlier in our place. It will always have a bond terminal, that is like covalently linked atoms terms, and there will be non-covalent interaction like non-bonded, etc. Sometimes even hydrogen bond can be an additional function. So it is a reproduction of my earlier one slide which says what is the form in amber force field. In that the form looks like this. There could be many parameters, and please do note all these parameters are dependent on the which force field you choose. We can use that. There could be some carbohydrate force field parameter which is included also, glycam. Now comes what is the difference between this energy difference and free energy difference. This to be remembered very well in your mind. Basically, free energy difference, which I emphasize that is important, and that is the measurable quantity like binding coefficient, etc., can be correlated with, or the energy due to the binding, it's a isothermic, that is it will produce a heat, endothermic, or it is an exothermic, all depends on that particular free energy difference. Let us understand what is free energy difference. Free energy difference, certainly it not energy difference. That's number one. But what it is actually, the term is expressed, the total energy difference distribution. It is a distribution function. So basically, the any kind of difference, any of the free energy is equal to the difference in enthalpy as well as entropy. Hence, it will be represented by the difference in energy at one point where two energy different distribution functions, that is rho A and rho B, intersects. What I mean by this is, if you think a system, that the systems are working, are having certain energy function in one rho A distribution, another have rho B distribution. We would like to find the difference between free energy by just having a overlap, that is the difference, the intersection of these two potentials. These two difference is the density distribution, or you may call it functions, rho A and rho B, is actually tells us how to do it. To construct properly these energy difference distributions, we require to go through all the possible conformation states in A and B, and must be sampled for reliable estimation of FBA. And that underlying reliable estimation is the most important. So obviously it tells us that there are chances that if we do not do proper conformation states coverage, we might not get exact or very reliable delta FBA. You will see, it will be seen in many cases, depending on the systems, how much we can cover the system. However, free energy difference will have all the components. Now, where we are at challenge. Challenge is sampling. That is how easily we can sample. As you see very simply from these two expressions written on the top, that the obvious is that we should estimate the delta BA across the board of all the states. So sampling of both states actually gives us the correct estimation of this delta HB, or free energy sampling. Trouble of sampling. Now sampling, one has to remember one thing. How is the sampling? What is the sampling? Sampling actually estimates ratio of partition function for each states. Okay. So what is the partition function or probability density, if you express, is given by this expression. Okay. And that is dependent on the delta. That's H itself, the H, energy of that state or free energy of the state. Now, if you look at the separable PR and rho PR, if we take the Hamiltonian with respect to and get a probability density of kinetic distribution. Kinetic distribution also will explain, almost will be explainable and integrated. So what we call these functions are, if we take it, a observable A in its radial distribution, or it's a function or averaging, average over the all ensemble, is represented or equal to by AR, that is dependent on R, PR, which is its momentum function, and DR. If we can integrate it over all the states, we get it a good Boltzmann distribution of a function A. Now, where is the problem of this configuration? Common sampling methods are available, like we should be able to just run the simulation on Monte Carlo algorithm and generate many particles, and they are different structures and different conformer, and we should get the average. Or we can run long trajectories and get lot of particles, different conformers, and then we collect them. We can calculate that values. So both methods are available to do it. However, it's not so simple. And why? The basic reason is what we call this space is quasi-nonergodicity. We assume all the time that if we run the trajectory sampling, that is, we run a system for a long under Monte Carlo or even in molecular dynamics, we will get huge amount of trajectory sample, which is something equation which should be that average of all possible configuration at one point of time should be equal to that particular expression, that is over a period of time. Now the question is, are they similar? No. Actually what happens, if you run for a long system or long time under equilibration, then what happens? You collect maximum number of configuration or conformation or structures or systems which are as a low energy stable. However, if there are transition state barrier kind of things or some states which are rather inaccessible by the macromolecule, least number of conformation you can collect there, however long run you do. Hence, this is where failure of ergodicity. So it is not the trajectory average cannot be really represent the average over the different conformer at one point. And that leads us to change the approach, how we change the approach so that we can cover the samples. So, sampling is difficult as I have shown you, especially to go to that level. Now, there are two ways of methods rather to elevate from quasi-nonergodicity. If we know the path, how from one to another I can reach, that becomes rather simple, and we can make and address these issues much easier way. Whereas when we don't know how to go to this higher energy landscape, where are many pits where there are isolated high energy barriers, it is very difficult to elevate that problem. So many methods have been discussed in future slides, and you will see them. When we don't have the knowledge about the pathway, how to find them for conformers so that they are really universally we can take the system. So enhancing this sampling when we do not know actually the conformation space becomes the most difficult part. So what we do normally is there are several methods, and I would just outline only every method, but I will talk about one or two methods only. One is, let us change the dynamics. That means what? So we sample across slow degree of freedom. That means where the motion of the molecule is very slow, we should search more of that conformer. And very quick, we will get lot of fast conformers. This is one. Another is sample configuration is changed because with a modified probability distribution. That means we can have a particular property which we add up to this potential so that it samples only those configuration which is relevant for our calculation, like collect lot of data at the transition state or at the hill point or higher point. Another is constantly we put another energy and or a function so that it deforms the energy space, so it actually increases the probability to reach the energy barrier quite fast. Means within our time fast. That means the time scale trajectory it will be accessing that. Another is using some forces. If we tweak the forces, there are multi-copy approach also are available, and there are other ways like using coarse grain methods, etc., where we do not use explicit everything. Like not if it is not necessary that every motion has to be trapped, then only the larger, slower motion, then we can represent them in a little coarse grain method. These are methods available, but I will show that two things. One is slow degree of freedom can be, is the, this is where the highest frequency of motion are related, and those frequency of motions where they're higher, they actually absolutely slow down the system. Hence, what we can do is we try artificially to put something so that the fast frequencies are damped, and the low frequency or slow frequency motions are compared to the first ones are actually predominant. That can be also to be done by certain ways, and one of the ways is using some master insert. I'll show you some example we have done, applied this. So I have discussed this. One can as well do another technique, and which is you use kind of hybrid Monte Carlo, or maybe using certain probability distribution using. However, what you can do is use a different time steps for encompassing the slow motion and different time steps for fast motion. It is obvious that the very fast motion you require a very low time step to follow it up. And on the other hand, to follow up the slow motions, you can tag with a different. This is another, of course, each one of them would require a lot of complicated or complex way of coding part of the protein and join together all the trajectory ultimately. Sampling enrichment is another way by doing the deformation of potential energy surface. Here I described about the this particular method, and I don't go detail of it because techniques are already been developed. But in principle, what we do is do not deviate too much away from the original distribution, but make it flattened or make it to have more and more sampling on the higher energy configuration than lower by using certain systems called hyperdynamics or accelerated molecular dynamics or something called poodle jumping, etc. These techniques have already been incorporated in some of the very nice new packages came up, and many papers have come to explain such and such methods. Enhancing sampling when we know the more or less the idea about the configurations space. So known configuration space, still it gives a challenge because we don't know really how the path is connected. However, free energy being a state function and it doesn't depend on the path, we should be able happily take any path and ensemble average can be done. So this has been shown to be very interesting, and one has done long back, our group itself has done long back this example, and you can see what I was meaning earlier, that how you can get ensemble average at lower level or low zero, almost zero energy very well and highly you can collect it. However, when it goes to the upper one, this dash actually shows how the function, when we collected enough amount of energy from that conformation, how it represented the energy. It was not smooth at all, and you can see that the smoothness curve fits only up to this, and the bad part you can very well observe on where it is on the top of the barrier because of not collecting enough amount of configuration. So this is a process we have used, actually umbrella sampling, and include them in calculate and find out more confirmation. Well, so there is another one, that is, instead of calculating probabilities and sampling across a whole surface, we would be interested to collect only at two states, let us say, not the whole path, but the two states. And that helps a lot if we know how to do this, combining two types of states and combine Hamiltonian. And if we calculate that, we can actually increase into two steps. We can have many steps connected. This is a simple equations how we develop that. So, which can be very handy, which is shown in the next slide. Definition of combined Hamiltonian is shown here. It can be dependent on a coupling constant or coupling parameter, which is called lambda. And this lambda is something, let us say we are trying to find a combined Hamiltonian, V combined, which is starting point, it will be VA, and ending point, it will be VB. And we will connect it with a function called lambda r, and it changes as we go from one state to another state, 0 to 1. So this can be explained by a few slides and is ideally using that combination of two states. We have actually displayed and took a system which is quite large system those days, that is Thermolysin, a big protein, and displayed free energy by calculate the difference between this free energy between these two systems by Perturbation MD method. The binding of phenylalanine, L-phenylalanine versus D-phenylalanine, and L-phenyl versus D-phenylalanine as a ligand to Thermolysin active site. And it's a large protein containing zinc, metalloion, and many other covalent bonds or coordination bond system. Hence, it was a difficult system. However, using this lambda parameter, which is shown here, associated with this dihedral angle, which is the improper dihedral potential, which is from one to another, A chemist cannot make this or believe this, but it is kind of what is called alchemistry. Computationally, we can do that. We can generate these, and instead of calculating this from going from phenylalanine water with thermolysin, we went only thermolysin with bound to L-phenylalanine. This bond is non-covalent. And we calculated, instead of calculating this system on the way that is called row-wise, we calculated column-wise. And that's where comes the most important thing of application of this. We use this potential, as I asked you earlier, that called chi angle 2 lambda chi 0 starting to end. And we yielded the result between the difference of potential or difference of free energy binding between the L-phenylalanine to D-phenylalanine. D-phenyl is preferred, where around 2.0 kilocalorie per mole, and we actually calculated the values which is shown here with a small amount of trajectories, and got it the values also minus 2. So L to D binding average is minus 2.0 and 1.8. This coupling parameter approach has been extended even more for calculating free energy, any kind of potential, and has been shown in few more slides followed. Detailed expression written here, which is of combination of two. We can expand it to more of Hamiltonian displayed here, and the energies and functions as such. How we do this sample? Most often what we do is tagging, importance sampling, or adiabatic decoupling. That talks about stacking. What we do in stacking? Actually, the replica exchange method, which has been quite popularly used, does this tagging or stratification or windowing of these independent different lambdas. The beauty of the system is you can actually generate all these configuration separately on your parallel computers and collect the density function and ultimately recalculate back your potential functions. Okay. Another method is to do umbrella sampling, which also likes tagging, but it will be a better approach and quickly can be done even if it is very high potential barrier. It is called adiabatic decoupling. It is nothing but this coupling is a lambda decouple motion. So what we are trying to do is we want to slow down the motions which are hydrogen associated motions, which is not very much playing any dominant role of this potential barrier, which is across some of the dihedral angle or torsional angle space. So to do that, we achieved this quite a long back, all the functions, and which is shown in this paper, and the plot shows that just as if the viscosity of the medium has gone down and gone slow. So this is an example of how to find the rate constant at different active transition states, and this is the application of an adiabatic decoupling. So we talk about here free energy estimators. Now generally sampling protocol, it uses a particular free energy estimator, but it is much wise to use many, because this can be applied also to sampling data. So common most free energy estimators is a global method, which is you actually count as many times the numbers the conformer has visited that place. Or another method is use the difference energy difference and calculate the FEP. Okay. Another method is local method, that is we just force this conformer and do the estimation, or one calculus actually adds up a potential associated with this transition probability and estimate that separately. So there are all these methods in different cases has been utilized. So this estimator decision is also a very crucial decision for estimate when we calculate the free energy. Now we come to the point is that when we have done enough sampling and we know the end state free energy, can we calculate that? That's the point. That is, we are not interested to really calculate all over the space the free energy, which seems to be quite a challenging job. However, can we calculate just the end state free energy? There are 2-3 methods. One of the method is a linear integration energy, which is some other people have really emphasized, and I will not discuss much here. However, there is the other method is to do with by MD simulation with either Poisson-Boltzmann or generalized bond surface area approximation. These two techniques have been discussed earlier while we are discussing about how to take explicit solvent effect in the system. First, I talk about linear interaction energy. It is more like snapshot of simulation represent the Boltzmann ensembles. And what we do is we average this over the electrostatic and van der Waals energies for the ligand with the solvent and the ligand with receptor and solvent. And then we calculate for few of them, find out what should be the function of alpha and beta. And then a good correlation with experimental value, we find that alpha is almost like 0.18 and beta is 0.33. And this function can be utilized for calculation of LIE type of methods. Many references are given in the top like this. In the text, all the references narrated by the same number. I would now discuss another important, which is more commonly recently are used, is a method called molecular mechanics by PB or GB assay. So these two techniques have been described earlier. However, for completion of my slide, I will show you those two formulas as well. Basically, what we are interested in is we collect many conformers over a period long time and collect these informations, and then we have used the same formula earlier talked about free energy. It is only estimates the EMM. So total difference of energy is expressed by EMM plus EG solve plus TS. However, what we are trying to estimate is EMM and G solve. Now, EMM comes from our trajectory of the main molecule, and G solve, G SASA and delta G ultimately comes from EMM plus this G SASA and plus this G PBIGA, that is the solvation. However, these 2-3 formulas which are shown on the right hand side does not include the T delta S. Hence, this method is very much applicable to systems which works not much influenced by the entropic motions or contribution from entropy. Generalized bond model, which includes spherical assumption of the solute and interaction with the solvents, which is shown in this formula here with the description. Poisson-Boltzmann model to interact with solvent and solvent-mediated electrostatic interactions. These two has been discussed quite well in previous modules. Here comes the final model. How we are calculating? We are just taking as if first we work on only ligand in solvation in solvent molecule, then ligand bound in the system. So we get certain values, that is the blue color contained the ligand. Then I used only the receptor and collect that. Then we put the ligand and collect the data. Now if we have different ligands, and we can calculate their difference of binding in vacuum and binding in solvent. Similarly, we can explain the binding in vacuum or both in water, but ligand 1 versus ligand 2. So this is shown very well in this particular picture and described. You can have so many interactions with the receptor and ligand. However, outside the active site, you will have only ligand and water interactions. So, as I have discussed earlier with about your what is the in principle to calculate the molecular mechanics based and with either GB or PB assay methods, I show you here a pipeline which has been developed by in-house with us, and it helps when you calculate many such trajectories and want to analyze it thoroughly. So this pipeline actually are in 3 phases as is noted, and takes care of many small problems or errors, and let you collect the amount of information what you want at the last level. You can develop one such pipeline before you start working on the systems as such. Here I just shown you the procedure how to set up in PB and GB in any one of those like amber force field, under that or any other force field you choose, but the NAMD software which gives you the facility to do this kind of calculations. Many other software has been integrated and developed. These softwares are developed by different people whose very the efficient programming techniques are written here in details, but mostly by Golike and others, but has been adopted in most of the software packages. However, one has to remember what should be the parameters to make it properly. Here I describe a case study using such pipeline and using only 4 very good, or I would say nanomolar inhibitors of a protein called kinase. This is called PK5 kinase. It is Plasmodium falciparum. So this is done in-house research. We had docked the compounds also by different methods as well. We tried to find because we know the binding activity from experiments, we tried to find their correlation. We also did use for all the 4 and the proteins as its molecular simulation trajectories to calculate the enthalpy by using MMPBSA and MMGB, both methods. On the top line row, you can see the crystal structure of the molecule. 2 are bound with one of the inhibitor and another one is a native protein. So 2 different inhibitors are bound with the structures are also seen. We do notice there are differences in structures. Hence, MMPBSA is expected to address some part of this lost energy or conformational change, what happens if we estimate it. Next slide shows us the binding inhibitors values, and how it is plotted is we plot for every index as shown in the box, small box. So we use DOC, we use GOLD, we use GLIDE and LibDoc. So you can see all the very good 4 softwares have been used, and the stars indicate the MMPBSA method. So you can see that try to show you the 1 sigma, how many of them are coming. So you would very simply say that DOC has performed very well. However, what we notice is introducing MMPBSA, it has given much better result within 1 sigma. Almost all the points except 1 or 2, a little bit borderline of 1 sigma. Otherwise, all of them has fallen on the 1 sigma value error, within 1 sigma error of all of them with experiment activities. So this shows you performance is very good. However, it also tells us maybe some of the inhibitors are not following just the enthalpic energy. So, as we saw, summary of the previous 2 slides is how one can find binding enthalpy of the results, and results are shown here. So difference of PB and GBSA, 2 types techniques have been utilized, and these 2 very interesting and highly, that is better ligand, who are really lower free energy ligands, have been found to be post proper place and found to be very much aligned with the calculation when we included exclusively the molecular mechanics using PB or using GBSA. This tells you that this ligand binding estimation of enthalpy based data could be the very ideally a very good way of finding instead of finding by docking. However, this also doesn't include that endpoint energy calculations of enthalpy, doesn't include the entropy contribution, and especially when there is a kinase which can move quite a lot, has been seen to be better affected by, strongly affected by the kinase, the motion of the molecule, and the motion of the ligand, then it is preferable that one should include the entropic contribution as well. So next few slides, I'll try to emphasize those, how one can go about to the entropy calculation. What is the conformational entropy basically? So it is called the Boltzmann entropy. This is proportional to the number of the system can occupy. And then if you look at the Gibbs entropy, which would be the way we would like to calculate this, would be that probability of microstate, if pi, then it will be minus kb summation over all the states possible. Hence, one can calculate very easily the conformational entropy, provided one can actually cover all the I steps. This is the challenge. I have told you earlier, sampling is also the challenge. This is also a kind of sampling. To calculate all conformational space, even which are higher state energy containing, would be very difficult. However, when we calculate the total conformational entropy, we require to use all the possible states. And that number is important. Hence, many methods have been developed almost last 10-15 years to estimate the entropy. That means as best the entropy can be estimated. There are like normal mode analysis, there are quasi-harmonic analysis, discrete molecular system, many is listed over here. We have been working on this problem for a long time and trying to estimate two methods at least, which I will take those two entropy calculations methods and display you few steps. What you should do if you use them. One is Accent-MM, another is Entropical. And I will compare their data at the end of it. However, as I mentioned, the system has to be chosen proper to calculate these methods. Here I show you a small peptide which is almost 20 mass, which is sequence is written here, which is in red color, which depicts the peptide which binds to Plasmodium falciparum AMA1. AMA1 actually is a apical membrane antigen. And the peptide, how it interacts is why it is biologically highly important. By this interaction, the Plasmodium enters the human host surface antigen and avoids the surface interactions and enters through it. Now this peptide, which is called Ron peptide, that's why it's named as R1, was missing one or two, but we modeled it here as you should see it. It has a not very structure, neither alpha nor beta, but it has a little turn in it. And this structure also has been elucidated in bound structure. What we are trying to find ultimately in the project is to how this entropy calculation affects the binding, which is certainly because bound and unbound structures are different. As well, the protein itself would be very different in that state. However, right now we have chosen this demo to do this only the peptide and calculate all the structures. Cluster validation, that is, many conformer, here is shown only the 4 very highly populated conformer and shown by this cluster analysis on the top, that these 4 highly conformer density conformers, they all look in different structures. So you can very well see, and they are stable structures because all of them are negative energy values, which is stated here. However, what we are looking for is how I can calculate collecting all the plots of cluster and utilize all these conformer. First, I do this clustering so that I know the major cluster where they occur, and then I include all of them, and then I calculate the parameters. As I told you earlier, it will be, we have to notice very carefully that the entropy contribution gets 1 point saturated. Otherwise, the trajectory is not sufficient. If I use the method called entropical, which is a balancing between certain entropy functions, here I show you the 3 plots. One is S1, that is only the first order entropy calculation, then second order value, and if we add up both of them, seems to be it is working much better. At least S1, S2 together has a better convergence. Both of them also has convergence. However, if you look at, you will see, if I take the enough frame and which has been shown here, only then the convergence is present here. What it tells you? You require to calculate a huge amount of trajectories to collect enough conformers which will represent every space it can traverse. That is the tough part of calculation of energy. This slide basically gives you the convergence of this mutual information of 2D by using entropical. Entropical is the method of calculation of entropy here for R1 peptide. And as you see, most of them really, peptide is really converging quite well. And I will discuss also about another method next, so that at the end we can compare with the two methods. Here I show you exact values of these contributions, and if you look at the convergence, you can very well find at the last few bins, and this binning is how you bin the structure. So if you take all of the trajectory and bin it in very haphazardly, that is arbitrary or random, you will never get actually the values. So one should do as much sample, as many bins where the sample lies. I have shown you only 4 cluster bins. Actually, it comes out to this particular peptide converges that almost 30 to 35 bins are good enough for going through. This may vary for different size of proteins. So all entropy values are kilocal per mole, which tells you with the different binning systems, all of them. Now I come to another method which I have used, and same type of plots are shown here because we would like to compare two methods. Here are two methods. One is accent, another is entropic. So you see two methods are used, and you can see both the methods gives reliably good at the higher number of bins. So take home messages. Number one, we must have huge number of conformers and long trajectory collections. We also must have distinct bins and large number of bins. Both is very costly, falls on CPU as well as on the hard drive. Here I show you that other method called Accent-MM method, by which also I want to show you that the most of the conformational entropies are converging. However, you can notice the last two are converging very slowly. And that only tells you the torsions are the most difficult to get proper convergence within, if we run very less amount of the trajectory and collect less amount of the conformers. The statistics of entropy I show here, and you can see it is quite an accurate values as you can find, both Accent-MM and the entropical. Entropical is actually more consistent in predicting the second order entropy with a varying number of bins, whereas Accent-MM is not so reliable. In the values are a little bit more, like almost 0.1 deviation. I come to the point now to discuss these efficient or very fantastic methods of calculations, which delivers a theoretician almost within the plus minus experimental errors, the values which are experimentalists can find, of course has a cost. Not only the theoretically system building and running efficiently the computer programs, it is the number of molecules if we are processing on this method of virtual screening kind of flow. This has been estimated in long back by a group of Samuelson and Aquist, and which shows this method accuracy and speed. Obviously, accuracy is better and better and better if we really do a pure free energy perturbation calculation. However, the speed of calculation would be really slow, and number of molecules feasible to process at the level of using it for free energy perturbation techniques, including enthalpy and entropy and everything, would be hardly 100. At the same time, you can process almost 10 to the power 6 number of molecules by using rather easier method called docking and scoring. Hence, question is how we balance it. It depends on, maybe once one should do the free all these docking etc., filter a few specific compounds and then do the thorough calculation so that you are confident what you are predicting is almost within certain 1 sigma error predictability is. So in summary, I tried to show you how to find the calculate free energy, which is a relevant important for biomolecular binding calculations and energy calculations and experimental data to compare with very many different methods, and elaborated on one method, that is endpoint enthalpy calculation, and that is feasible mostly at present with our computational ability. However, free energy includes the entropy, not only the enthalpy, and in cases like kinase or proteins which modulates themselves during the binding, it's very important to include the entropic terms. However, I have shown you with some of the examples as well, the entropy calculation is really a heavy duty calculation and on the CPU as well as data, that is storage of data. So many times we omit the entropic part and we just assume that while binding or free, the entropy contributions are not so much important, which is not a correct estimation, as we find more and more new proteins and many protein structures are elucidated. And when you do this comparative study, I have also tried to show you with two examples where I have shown you how you can compare your data with experimental as well as theoretical simulation. Thank you.